Hi all, my name is Alda Cahyaningtyas and this is my video submission for microbiology for module 2. To begin this video, I'm going to talk about the formation of biofilm. Generally speaking, it takes at least 6 steps for biofilm to be formed. The first step of biofilm formation is the presence of free living cells or microorganisms and surfaces. The surfaces can be anything, it could be non-living thing and could be living thing. It could be a tooth, it could be the floor of your shower, could be the shower curtain. Once there is contact between the microorganisms and their surfaces, the cells will react through metabolic response. And this is the second step of biofilm formation. In order for the metabolic response to take place, obviously there must be some kind of receptor to the stimuli of being in contact with a surface. And the receptor or the form of receptor in this case would be for some bacteria it would be the fimbriae, and in some other bacteria it would be the inner component or inner part of flagella. Now we're able to move on to the step 3 of biofilm formation, where the cells create this gooey, sticky, slimy complex, and it is called extracellular polymeric substance or EPS. EPS is composed of DNA, polysaccharides, and proteins. Due to the physical characteristics that EPS possess, the cells now have the ability to attach to other cells, to attach to a surface or substrate, to isolate nutrient or have nutrient intake, and create water passages or channels among the cells themselves. And then they also enable the cells to create their own community. It's like microenvironments of the cells within the biofilm itself. The last but not least ability that now the cells have, because they have this biofilm, is that extra protection against environmental stresses. The environmental stresses come from different sources. They can come from pH, temperature, humidity, antimicrobial drugs, and also UV radiation. So now, because they possess the biofilm, they have more resiliency, they have more resistance towards these environmental stresses. Now I'm gonna continue to step number 4, and also I'm going to speak about quorum sensing and how they relate to the biofilm formation. Quorum sensing is a process. It's a process in which microorganism is able to respond to the density of microorganism surrounding them. Hence, in step number 4 is when the microorganisms secrete quorum sensing molecules that will count or communicate the number of microorganisms surrounding them, and also detect or identify the type of microorganisms within or among the biofilm. Step number 5 of biofilm formation. So the quorum sensing molecules act to count or communicate the number of microorganisms surrounding one particular microorganism. So when there are more microorganisms, the cell itself will secrete more and more quorum sensing molecules. When both of these numbers go up, um, the microorganisms have receptors to bind with the quorum sensing molecules, which ultimately lead to more and more binding of these receptors and quorum sensing molecules. Eventually, a threshold of number of binding is going to be reached. And when that threshold is reached, the cells will activate or turn on previously suppressed genes. When these previously suppressed genes are activated, this leads to new traits. The new traits that the cells now possess will work in favor for the microorganisms and for the formation of biofilm. So the new traits that could come up are secretion of new enzymes, the changing of the shape of the biofilm itself, and then instead of pushing away new microorganisms or um, diffusing them away, um, they will encourage other microorganisms to kind of um, go around them to create a more complex biofilm structure. So when new microorganisms arrive, they have this relationship, this new synergistic relationship with the pre-existing microorganisms. Um, they sort of assume different roles for the microenvironment. So it's like a community of different microorganisms, and then they now possess new roles that otherwise they wouldn't have if they were to function as a single cell or a single microorganism. And um, just like a typical community, some members of the community may want to leave. So what's going to happen for step 6 is where some cells or some groups of cells leave the biofilm, and they leave in the form of streamers. And once they leave, these microorganisms, these cells, are able to um, go through the cycle all over again. They can attach to another part of the surface to create biofilm, and basically go through the entire cycle again to give um, more advantage towards the microorganism, but obviously would be very disadvantageous for the structure or body that's being attached by the microorganism through biofilm. This continuous cycle can go on and on and on and on, non-stop, and this goes to show how persistent and how resilient microorganisms can be. And it is also important to understand that once microorganisms have formed biofilm, they're actually more harmful. Since we know that microorganisms become more harmful when they already form biofilm, a lot of researchers are finding ways to prevent the formation of biofilm in the first place. One of the ways that they have um, found um, is actually inhibiting the receptors of the cells that are supposed to bind with quorum sensing molecules. Since um, the quorum sensing molecules are not able to be bound with the receptors, the cells are also not able to create new traits, leading up to them being more resistant by forming biofilm. So this is definitely an effective way of preventing the formation of biofilm. But then what happens if the biofilm has already been formed? Um, in some cases, other researchers already also found a method of disintegrating the EPS or the biofilm um, through enzymes. So these enzymes will essentially destroy the EPS and allow antimicrobial drugs to work against the microorganism. Uniquely, some microorganisms do not form biofilm when they're only in a small number. So what they do is they hide in capsules or in blood clots, and because of hiding, they become undetectable by um, the host immune system or our body's immune system. So, what the researchers do, they artificially give quorum sensing molecules. So, when there are an influx of quorum sensing molecules, obviously the receptors of the microorganisms want to bind to the quorum sensing molecules. This reaction of wanting to bind to the artificial quorum sensing molecules enable the immune system to detect the small number of microorganism, and hence um, triggering the actual response of immune system and eventually killing the microorganism.